Welcome to the Ticktective podcast and video series, a program of the Bay Area Lyme Foundation, where our mission is to make Lyme disease easy to diagnose and simple to cure. I'm your host, Leah Gertner, Director of Education and Outreach. Lyme disease is the most common vector-borne disease in the USA and is a global issue. This show includes interviews with the researchers we fund, as well as other interesting people in the Lyme scientific community. We are a nonprofit foundation based in Silicon Valley. Thanks to a generous grant that covers all of our overhead, 100% of all donations go directly to support research and prevention programs. You can find out more or donate at bayarealyme.org. Welcome, Dr. Jacob Lemieux, to the Ticktective podcast and video series, sponsored by the Bay Area Lyme Foundation.
Thanks for having me.
Jacob Lemieux is a postdoctoral researcher in the Sabeti lab at the Harvard-MIT Broad Institute. Dr. Lemieux also just completed an infectious disease fellowship at Massachusetts General Hospital and Brigham and Women's Hospital. He holds a bachelor's degree in science from Stanford University, a doctorate in clinical medicine with a focus in molecular parasitology from Oxford University, where he was a Rhodes Scholar and member of the NIH Oxford Graduate Partnership Program. And he also holds a medical degree from Harvard Medical School. Congratulations on finishing your infectious disease fellowship at Harvard and becoming junior faculty at Harvard Medicine starting tomorrow.
Thank you. Yeah, it's been a lot of education and it continues, but very interesting and a lot of fun.
I know you studied malaria at Oxford, and I understand that you've moved into tick-borne diseases such as Babesia and Borrelia at Harvard, and I know that malaria and Babesia are somewhat related, and I actually have a quote here from Oxford Medicine that says, "Malaria parasites and Babesia both inhabit red blood cells during part of their life cycles, and these stages cause the diseases malaria and babesiosis, which are similar in many respects. The facts that humans can occasionally acquire malaria and babesiosis from animals, that both parasites appear similar when seen in blood films, and that both cause similar symptoms can cause problems in diagnosis in these rare infections and are therefore of interest to clinicians and epidemiologists." So I'm wondering if that is how you got started in tick-borne disease research, jumping from malaria at Oxford.
That's exactly right. The first time I heard about babesiosis was actually when I was working in Kenya during my PhD on a malaria project, and we had a seminar from somebody who was talking about antigenic variation in babesiosis. And I said to myself, what's babesiosis? And I learned that it was a cow parasite that had some similarities. And then I didn't encounter it again for a number of years. But when I came to Boston, somebody referred jokingly to quote unquote Massachusetts malaria. And I started asking about this parasite and reading up on it, and it turned out that it really wasn't well studied. And so I came to Boston to study medicine as a malaria researcher. I had no intention of switching, but I picked up a new interest in babesiosis and became fascinated by this largely unexplored area of human parasitology. And I stuck with it after encountering on my clinical rotations a number of cases of babesiosis. I remember the first one was a patient with a persistent fever, and we couldn't figure out what was going on. So I went down to the microbiology lab to look at the peripheral blood smear, which I had done many times during graduate school. Was quite used to looking at those from malaria. And there were parasites on the smear. And it was incredible to be able to make the diagnosis in that fashion. And from then on, I was hooked, and we put together a project to study Babesia genetics. It was largely unexplored. I have a particular interest, as does Paris, who is the principal investigator in the Sabeti lab, in genomic sequencing. And so we did some of the first genomic sequencing in babesiosis, and that produced a number of exciting insights about population structure and relapsing babesiosis in a subset of individuals. So we stuck with it and then started to look into other tick-borne co-infections. And it became clear that this is a group of diseases because of shared ecology and epidemiology and clinical manifestations that should be studied together in some sense. So that's how I've come to Lyme disease research and tick-borne disease research. But I do still really find malaria interesting. I'm not sure I've completely given it up, but right now I don't have any active research projects on malaria.
Well, a win in Babesia might end up being a win in malaria, right?
That's the philosophy that I take, which is that intracellular parasitism in human red blood cells, and red blood cells in general, is an infectious niche. There are particular types of patterns that are reproducible and observable in these types of infections. The patients are sick in some of the same ways. So there are some similarities and there are some differences, and they're meaningful, and they help us understand how these parasites cause disease, and they may identify areas where treatments can be more effective.
So congratulations for winning the Bay Area Lyme prestigious Emerging Leader Award. There are only a couple of these given each year, and your research holds a lot of promise. I'm wondering if you can tell us a little bit about the project that you proposed to the Bay Area Lyme Foundation.
Absolutely. One of the challenges with Lyme disease is that the diagnostic tests rely on a person's immune response to parts of the Borrelia antigens. And so the Lyme diagnostic assays have two particular shortcomings. They don't work early, and they don't work in patients who have immunocompromise or immunosuppression for one reason or another. They don't work reliably in those circumstances. The alternative would be to have an assay that directly detects bacterial products. The challenge has been that culture of the organism and PCR detection of nucleic acid from the bacteria, which are the two mainstays of microbiological diagnosis, have had low sensitivity for Borrelia burgdorferi detection. In addition, culture is technically challenging to perform. It's much more complicated than the usual type of bacterial culture that could occur easily on a petri dish or in a broth in a lab. So there are some key technical challenges. We would prefer, all other things being equal, to have a test that directly detects the products of an organism, because it could work earlier, it could work in a larger set of patients, or it wouldn't stumble in those subsets I listed earlier. And in addition, one might be able to use disappearance of bacterial products as a way to demonstrate successful treatment of a patient. For example, that's what we do in malaria or babesiosis, where we count the number of parasites or the percentage of parasites that we see in the red blood cells. And as those parasites go away, we are reassured that we've had success in treating the patient. So it would be highly desirable if we had an assay like that for Lyme disease. However, many people have tried and not been successful. But recently, there have been some findings that suggest it may be possible to make some progress in this area. And those findings have largely centered around the fact that if you increase the volume that you use for Borrelia culture, you can increase the yield to a place where you can detect Borrelia burgdorferi bacteria in the study that I'm thinking of here in over half of the patients, if you use a combination of culture and PCR, approaching 70% among patients with acute Lyme disease. In addition, there have been studies led by Dr. John Branda and Dr. Mira Pollack, one study that I was a part of, where in the majority of patients using shotgun genomic sequencing, patients with confirmed acute Lyme disease, the group was able to see detectable nucleic acid in the blood of these patients. So there are signs that the bacteria in certain circumstances may be present in a majority or even larger portion of patients, and that if you use the right approach, then you may be able to detect it. So it's really about technically optimizing the conditions for detecting the bacterial products. And one of the advances that we would try to use is whole genome shotgun sequencing. That allows you to detect the whole bacterial genome rather than just a fragment of the genome, as is done in conventional PCR. The problem with that is that you detect all nucleic acid, and there's a lot of background nucleic acid from the host. In this case, that refers to a human. So there's another set of technologies that allows you to selectively enrich the microbial product, the microbial nucleic acids from the background. And that allows you to amplify the signal. So using whole genome sequence, we have a larger set of things that we're potentially able to detect. And using hybrid capture or target capture, we're able to enrich for the things that we care about. And it doesn't just have to be Borrelia, it could also be other tick-borne pathogens, which may occur in co-infections. So this would be a way, we hope, to test for the presence or absence of multiple tick-borne pathogens at one time and to boost the signal to detect a number of tick-borne pathogens, particularly Borrelia burgdorferi, which causes Lyme disease.
Right, and I know some of the tick-borne diseases that you were looking at are babesiosis, anaplasma. I know Powassan virus, was that in there?
The first thing that we're going to use is we're going to start with a technology that we developed and tested a number of years ago. In 2016, we published a paper on this where we developed a target capture method for 5 tick-borne pathogens: Borrelia burgdorferi, Borrelia miyamotoi, Babesia microti, Anaplasma phagocytophilum, and Powassan virus. The only one we've extensively tested it for is Babesia microti. And we plan to do more testing for all of them. We may even modify the enrichment method to include additional pathogens or to change the sequences that we put in there. Powassan virus is important because it causes serious disease. We don't understand how common it is, but it also brings a unique challenge that's an RNA virus. And so we have to do an additional step of changing the RNA to DNA before we process it in the same way. So we're very interested in Powassan virus. I think we plan to investigate the feasibility of including it in this approach, but I think the major focus of this project is Lyme disease and the other co-infections, Borrelia miyamotoi, Babesia microti, and Anaplasma phagocytophilum.
Great. Well, Lyme disease patients all over the world will be very grateful that you're focusing on this understudied and under-researched and underfunded disease. I know that you are collaborating with our Lyme disease biobank, and I'm wondering if you could speak a little bit about your collaboration.
Yeah, so we've collaborated with the biobank for a number of years. Most of the collaboration has been around us providing samples to enrich the biobank. We have in our collections an interesting caseload of patients who may have more severe manifestations requiring hospitalization. And given our prior interest in babesiosis, we have a number of patients who have babesiosis, sometimes with severe complications. And so the lack of availability of samples to researchers, I think, is a major barrier to high quality Lyme disease. So what the biobank is doing to address that challenge is really important, and we try to support that as much as we can. So we've had a productive collaboration around enrolling for several years. But in this project, we're also able to access samples from the biobank and study those. And we did a pilot cohort as a part of Dr. Sabetti's Emerging Leader Award, focusing on whole genome sequencing to study the evolution and origin of Borrelia burgdorferi. We did a pilot project and found that we were doing a great job detecting babesiosis in those pilot samples and doing a reasonably good job of detecting anaplasmosis and ehrlichiosis, although the numbers were small. But it also made it clear that we were doing a poor job of detecting Borrelia burgdorferi without some additional optimization. Studies in the literature have shown that in the cases where one can find Borrelia burgdorferi, the numbers are extremely small in terms of bacterial load in blood. So one thing that's clear is if you're looking for a rare amount of organism, if you start with more input material, that can only help. And so we want to start with as much as possible, but of course there are limits on this. There are limits based on the size of a blood draw that it's safe to conduct in a human. Even if it is safe, people are not always interested in having a large volume blood draw. So there's some practical considerations. And then in terms of attaining that large of a sample, there are also considerations about how to process that to extract the nucleic acid from them. So one of the goals of our work is to find the sweet spot where we get the right amount to maximize the yield of the diagnostic, but we also don't continue drawing extra blood or using extra material if we're in a place where additional blood means diminishing marginal returns in terms of our ability to detect the microbe. So investigating that is not particularly flashy, but it's critical to figure these things out. What's the right volume of blood? What's the best way to process the sample? How is this affected by various extraction methods? those types of things will influence and will set you up to successfully detect the bacteria in the downstream detection steps, which in our case involves sequencing.
And in your training as an infectious disease fellow, did you get ample training on tick-borne diseases?
We got a lot of exposure to tick-borne diseases because of the environment and the part of the country that we're in. So we got a lot of exposure. There are many unknown questions. For example, during my training, Borrelia miyamotoi disease, Borrelia miyamotoi as a bacteria, both of those things were newly appreciated. And people had known that this was a bacteria for a number of years, but the fact that it caused disease in humans was not appreciated until just a few years ago. And Powassan virus is similar in that this virus has been around for a long time, but it clearly had an uptick in this part of the country during the time that I was in training. And perhaps some of that is us recognizing the disease more. Perhaps some of this is a true increase in prevalence of the disease in the population. We got a lot of exposure, but part of our job as investigators is to learn more and to keep studying the diseases that we begin to encounter, to learn new things about the diseases that we have seen for a long time, and then potentially to recognize new disease entities. So that's one of the best parts of the work, is to be able to participate in this type of research.
Do you have a sense of why research on these tick-borne diseases has lagged behind research in other disease areas?
The challenges of tick-borne diseases is they don't grow well in culture. And culture has been the mainstay of diagnosis for a long time. And it's been the mainstay of producing enough material for laboratory investigations. But Borrelia burgdorferi, while it can be cultured, is not easy to culture. The same with Borrelia miyamotoi. Babesia microti cannot be cultured. Anaplasma phagocytophilum is hard to culture. And Powassan virus can only be propagated in the lab under high biosecurity, biocontainment conditions. So they're not easy to work with at that level. I think that's made it harder. That's probably the major thing. There's also been a sense that many of the tick-borne diseases cause similar clinical syndromes, and that doxycycline can just be applied as a blanket treatment. And doxycycline is effective in a number of situations, but there are some, like babesiosis, where it doesn't have any activity. And so I think that in part because the diseases are hard to study technically, the diversity, both microbiologically and clinically, of tick-borne diseases has not been fully appreciated. This is an area where molecular diagnostic methods are really in a good position to both facilitate research, but also potentially assist with diagnosis.
As a physician who is about to go into teaching, do you have some thoughts on how to bring tick-borne disease more to the forefront for diagnosing physicians around the country?
I think it really gets back to good diagnostic thinking and diagnostic fundamentals. As physicians, it's particularly important to recognize that there are many things that we don't know, remain humble about what we don't know, and try to search for the answers. I think this applies in all of medicine and in tick-borne disease. I think it's been humbling to see that there's newfound recognition of a number of new pathogens over the last few years. And it's just a reminder to all of us that the textbook of medicine is not complete. So we continue to learn things, and diagnostic reasoning is a critical clinical skill. And it has to recognize that there are many things we don't yet know.
Well, we're learning that with coronavirus right now, aren't we? There are new pathogens coming into the medical realm constantly. And I don't know if you've been doing research in the lab there on the coronavirus, but I'm just interested in hearing what sort of interactions you're having with that virus at this point.
Well, we've been deeply engaged in coronavirus research, and the hospital has been deeply involved in care of coronavirus patients. So all of us have been trying to assist in various ways. But in the lab, the thing that we've been doing the most is just to apply our expertise in diagnostic methods and genomic methods to coronavirus. And so we were involved early on to develop diagnostic assays for SARS-CoV-2, and now we have a big effort to do sequencing of SARS-CoV-2 to understand the way in which it spread and the epidemiology of the virus. So I think the nice thing about the work that we do is that it can be applied to a number of different pathogens and infectious diseases. It produces a kind of information that enriches our understanding of clinical manifestations of disease or traditional epidemiology.
Can you tell me a little bit about your thoughts around coronavirus and Lyme and the intersection there in your world?
When you're sampling nasopharynx of a patient with COVID-19 or the oropharynx looking at saliva, you're focusing in on the site of the primary site of disease, which is the respiratory tract. And so there are billions of copies of virus there in many patients. Whereas for Borrelia, you're looking for 1 or 10 copies in a 1 milliliter specimen. So there's several orders of magnitude difference. It's just a much harder detection problem. But that doesn't mean that it isn't impossible. I think we don't know the fraction of the time that there are any Borrelia in the bloodstream. So it's somewhere between 0% of the time and 100% of the time. Patients with acute Lyme disease will have blood that contains Borrelia. If almost all of them have it, then it's a detection problem. But if the percentage who have any bacteria in the blood is less than that, then it imposes an upper bound on how sensitive a diagnostic using this approach, which is aimed at trying to detect directly bacterial products in the bloodstream, could ever get.
Because in the early stage, the Borrelia, like you said, might be found in a skin sample if somebody happens to get an EM rash. But in the later stages, the bacteria are hiding in more avascular collagenous areas. That's what you're referring to when you say it might not be in the blood at all in some patients, correct?
What I'm saying is when the patient is bitten by a tick and the patient develops Lyme disease, the greatest concentration of bacteria tends to be in the skin around the bite site. And we're looking in a place that isn't ideal. In patients with late manifestations of Lyme disease, for example, Lyme arthritis or Lyme meningitis, detection of Borrelia is similarly challenging, and you really have to go looking where the disease is. For Lyme arthritis, there's actually pretty good sensitivity of detecting Borrelia nucleic acid in the joint. For Lyme meningitis, the sensitivity of PCR has historically been much lower. So it's a challenging problem because we only sample particular sites and bacteria may or may not be there depending on timing and the type of disease being caused.
Well, if you have Lyme carditis, you're in a bit of a pickle. If you have neurological Lyme, you're in a bit of a pickle. We don't commonly sample the brain and the heart, right? So if it's not free floating in the bloodstream, but it's taken refuge in one of those areas, that could probably be a tough diagnosis, right?
Absolutely. We don't routinely sample those sites for any diagnosis. I mean, there are situations in which we do biopsy the heart or the brain, but only when the risks are worth accepting in those situations. And those types of situations are extremely uncommon that we would be willing to take the risk of biopsying somebody's heart. Only in cases where there's progressive injury to the heart muscle causing progressive deterioration in the patient with significant diagnostic uncertainty. Those are the only situations in which those types of tissues are sampled. But I will say that they do happen occasionally. I think one of the advantages of having a target capture method is that it allows us to look in samples that have been collected for other purposes, whether we can find any bacterial products, bacterial nucleic acids in those tissues.
Which would be different than PCR.
Yes, because it would allow us to look for a larger number of sites in the genome. The limitation of PCR is that you look at a particular fragment of the genome, and if that fragment is not present in the sample that you have, you may not see it, or you won't see it if it's not present. But perhaps just half the bacterial genome, or perhaps just one plasmid from the bacteria happens to be present in that sample. You'd have a much better chance of finding that using whole genome sequencing, particularly if coupled with target enrichment. So we think it might be useful as a diagnostic tool, but An added benefit would be that if we can kind of get this thing working and more widely distributed to other labs, that it could have important implications for research.
Such as?
It would allow us to look for the presence or absence of bacteria in ways that might be more sensitive than PCR. And it would also allow us to ask whether things like, does the particular Borrelia strain that you're infected with influence the clinical manifestations that a patient experiences? So it's an open question whether some strains of Borrelia are more likely to cause arthritis or carditis. It's challenging to do that research because strain typing is typically only available if you can culture the organism. So it would be really valuable if we could strain type by sequencing without having to go back in time and be able to put the sample into culture media to keep the bacteria alive, that if we could reconstruct this from nucleic acid. So that would allow us to use some of the techniques like bacterial genome-wide association studies that have been used for other bacteria for Lyme disease.
That is really exciting for us who've been working in the Lyme diagnostic world for a long time. I'm wondering if in late stage or persistent Lyme disease, if you have thoughts on the causality behind that. I mean, of course, there are many factors and nothing is certain, but I'm wondering when you're talking about strains, if you have any theories about that.
I personally don't have many theories. I tend to be just a let's collect the evidence and test hypotheses kind of a person. So look, there's a lot of historical controversy in the field of Lyme disease, and there's a lot of animosity between particular groups or individuals. I want to focus on the concrete research barriers that we might be able to make some progress on so that we can take better care of patients. And I find the literature on Lyme disease fascinating. I think it raises more questions than it provides answers compared to other fields of infectious diseases. The studies of Lyme disease are small. There has been a paucity of clinical trials performed. There are few studies that have used microbial culture along with detailed investigation of clinical outcomes. So I think there are big kind of gaps that it will take lots of collective effort to fill in. But those are things that I'm excited to try to work on in collaboration with other investigators and patients who are interested in research.
Well, thank you so much for joining us. I'll let you get back to work, back to the lab, and back to treating patients. Is there anything else you'd like to add?
I appreciate all of the support from your organization. And as always, very grateful to the patients who participate in research studies. I think is a critical collaboration between researchers and patients to work together to produce new insight to help us diagnose and treat diseases better. So thank you. I look forward to keeping you updated on how things go with the award. We're really excited to carry out the research and see what we find. I'm optimistic that we'll be able to make some progress. I think this is a big challenge though, and I'm not sure that we'll be able to crack this nut in a one-year research project, but I think it's just a longer-term project that needs effort from a number of different angles, and so we're really excited to get started.
Thank you for joining us for this episode of the Ticktective podcast and video series, a program of the Bay Area Lyme Foundation. You can find out more or donate at bayarealyme.org.